Following on from our lesson, action potentials and transmission of action potentials along an axon of a neuron, today we are going to continue at looking at the transmission of electrochemical messages. We're going to look now at how these messages can travel from one neuron and jump to another. Now, the name of this process where we jump from one neuron to another is we're jumping across a synapse. So let's start with some key definitions for today. Firstly, again, that word, synapse. A synapse, very simply, is a gap between the axon terminal of one neuron and the dendrite of another. This is very similar to a neuromuscular junction. And a neuromuscular junction, again, is a gap between the axon terminal. However, this time, a slight difference is it is a gap between the axon terminal and what is known as skeletal muscle, an effector. The third and final definition that's important for our lesson today is neurotransmitter. And a neurotransmitter is a hormone that carries a nerve impulse across a synapse. So let's have a look at a diagram and see what this actually looks like in reality. In this diagram here, we have two neurons. My first neuron over on the left-hand side, you'll see I'm colouring in all of the axon terminals leading back towards the axon itself. And in the second neuron, in the green highlighter, I'm highlighting all of the dendrites over on the right-hand side of this other neuron. Now, these two here connect, but they don't touch, okay? There's a very small gap known as a synapse in blue, and if we zoom in really closely, we can see this gap in between these two. So this blue gap is the synapse. So again, I'll colour in yellow our first axon terminal, and this connecting to the dendrite of the next neuron. Now, if we look really closely in this synapse that I've highlighted in blue, you can see also these neurotransmitter molecules and neurotransmitter receptors. Now, they together will pass on that message. So if we have a scroll down now and we have a look at this diagram, we can see a little bit closer and a little bit clearer what's happening in a synapse. And there are some key things happening here. Number one, the synapse, also sometimes referred to as the synaptic cleft, is this region in between these two neurons. Okay, so that there is the synaptic cleft. The next key part is the knob, and this is simply the knob of the axon terminal. Okay, so the end of the axon terminal flooding into that synaptic cleft. The third key part, represented in red on this diagram and contained within these vesicles, is neurotransmitter. That neurotransmitter is what is going to send that message from the axon terminal across to the dendrite of the next neuron. On the other neuron, on that dendrite, so if I highlight my dendrite in green here, similar to our last diagram, this dendrite is attached to the next neuron, and attached to the dendrite we can also see these small receptor molecules. And if I highlight some of them in purple for us here, we can see the receptors on the surface of the dendrite. So what actually happens? How is a message sent from this axon terminal to the next dendrite? There are 5 key steps. The first step, I'll use a different colour to represent each. A nerve impulse arrives, and we can see this impulse arriving at the end of the axon terminal. It arrives at the synapse. And what this does is it causes calcium channels to open, and this results in vesicles being traveling towards the membrane. So these vesicles filled with neurotransmitter are gradually heading towards the cell membrane, towards that synapse. Now, as they get closer, eventually these vesicles merge with the membrane. And what that means is that they then empty themselves and they empty the neurotransmitter into the synapse. And we can begin to see that in what I'm highlighting in green now, this neurotransmitter being released into the synapse. Step 3 in this process, and there's an important use of a word here, that neurotransmitter diffuses across the synapse. And by diffusing, we mean that it is traveling across the synapse. And we can see this neurotransmitter unloading and moving across the synapse here. it will continue to move across that synapse until in step 4, it binds with a receptor. Oh, apologies for the colour there. It binds with a receptor on the adjacent dendrite, and we can see that in 2 instances here that I'm highlighting in purple. Now, once the neurotransmitter has bound to that receptor, it then will result in sodium channels beginning to open or depolarization, and that gradual depolarization will then lead to the commencement of another action potential. So there are 5 key steps. So to very quickly recap, number 1, a nerve impulse arrives at the synapse, and this leads to calcium influx into the cell, leading to these vesicles moving towards the membrane. When the vesicles eventually make it to the membrane, they merge with it, and the neurotransmitter is emptied into the synapse. Once the neurotransmitter is in the synapse, it diffuses, it moves across the synapse to the receptor on the adjacent dendrite, where sodium channels are told to open, and this leads to depolarization. So there are a few other images that you can have a look at throughout the slides. However, we're going to look at 2 other things with this lesson, so only 2 slides to go. The role of enzymes in the synapse is one of them. So, when we're talking about synapses, something we've neglected to mention so far is that enzymes are found there. And their role is to help break down neurotransmitters to either stop the transmission of signals, okay, or to ensure that we don't produce an action potential in the postsynaptic neuron. And another reason that this can happen is because these products want to be recycled and we may not want to send a message, okay? So we can stop the transmission of these signals so that there is no production of an action potential. We're trying to prevent that. It does not continue to produce action potential. The second and final, so the final thing here, when we're talking about some of the chemicals that can affect synaptic transmission, there are 2 broad categories. The first of which are stimulants. So stimulants are things which increase the activity of neurotransmitters, and they can do this either by promoting the release of neurotransmitters or maybe by preventing them from being broken down and reuptaken by the cell. And this can lead to a period where the neural signal or the nerve impulse is constantly being fired because these receptors on the postsynaptic neuron are constantly being triggered. Depressants, on the other hand, so things like anesthetics and some venoms, can decrease neurotransmitter activity. And they can do this by either blocking the release of neurotransmitter or enhancing how quickly they're reuptaken. And this can result in the opposite effect. So reduced neural signaling, reduced stimulation of the postsynaptic neuron.